Hello, everyone, and welcome to today's session. My name is Marta De Gia. I am a SYNGAP parent and director of SYNGAP Research Fund. We are very excited to continue the SRF SYNGAP Research Fund webinar series. The goal of the series is to empower your communications with clinicians as you get more clear knowledge of SYNGAP. We also want to give you a plug to our next presentation, Treatments in Development for Epilepsy Syndromes, Opportunities for SYNGAP1, which will take place on Thursday, September 2nd, 10 a.m. Pacific time and 1 p.m. Eastern time with Dr. Anna Mingorance. Our talk today is Interpretation of SYNGAP1 Variants. I have the pleasure to introduce today's speaker, Dr. Eduardo Pérez Palma. Dr. Pérez Palma is a bioengineer and a doctor in molecular bioscience. After finishing his studies in Chile, he did a post-doctorate at the Genomics Center at the University of Cologne, Germany, funded by the Dravet Foundation. Since then, Dr. Pérez Palma has specialized in the genetic study of patients with epilepsy and neurodevelopmental disorders, focusing on the development of tools for interpretation of variants. He was a research associate at the Cleveland Clinic until last year, and now he is at the Center for Genetics and Genomics at the University of Development in Chile. A recorded version of this webinar will be available on the SRF website under webinars on the family menu. By the end of the presentation, you will have the opportunity to get the answers to your questions. We love to hear from you. Please write your questions in the Q&A. For those of you just joining us, welcome. And again, the speaker is Dr. Eduardo Pérez Palma, and the talk today is Interpretation of SYNGAP1 Variants. It's now my pleasure to turn things over to Dr. Palma. Welcome.
Thank you, Marta. It's my pleasure to be here today, and thank you also for that kind introduction. Well, as previously announced, the title of my presentation is called Interpretation of SYNGAP1 Variants. And well, the contents of this talk, first, I'm going to introduce to you two basic concepts about genetic variation and SYNGAP1. Then I will continue with variant interpretation concepts, the methodology of variant interpretation, and then to describe to you some complementary methods that we can input to this process and help to aid in the identification of pathogenic variation. Then I will continue with an example of how to interpret SYNGAP1 variants, and then some ideas and concepts that will try to describe to you where is this whole field going and future projections. So first, let's start with genetic variation and SYNGAP1. Genetic variation in the human genome is highly normal. Between two individuals, you can expect between 4 and 5 million nucleotides involved in genetic variation. Among these, any normal individual with no apparent disease will have between 150 and 200 protein truncating variants, which these ones are usually diagnosed as pathogenic variants, but naturally we have between 150 and 200 of these events. Also, the most common type of variation is by far missense variation. And here we can expect in normal individuals between 10,000 and 12,000 missense variant events. Of all of these genetic variants that one will carry, between 40 and 80 events will be de novo. That means that it will be absent from both of our parents. And among these, 1 or 2 will be on the coding portion of the genome. That means the genetic code that leads to protein production. These numbers tell us the first message that any genetic test is going to find variants. This is going to happen. The key question here is which ones are the ones causing the disease? Which are the ones that are pathogenic variation? We have many types of genetic variation. We can expect in every gene in our genome to have at least 2 copies, one from our mother and another one from our parent. Missense variant change a single nucleotide that leads to a change in an amino acid within the protein sequence. This may or may not lead to a change of function. We have also protein truncating variants that stop the protein production and then usually leading to an haploinsufficiency. That means because we have only just one copy of this gene. Also, we can observe within the human genome full gene deletions, full gene duplications. The functional effects of this variation, it's very variable too. We have gain of function and loss of function from missense variants. We have mostly loss of function variation within protein truncated variants and from deletions. Also from duplications, we can expect gain of function because we have multiple copies of the same gene. Of course, in biology, there is an exception for everything. So if you go through the literature, you can find exception for each of these cases. Missense variation by far are the most common type of variation and also the most difficult to predict. If we go a bit more in deep into genetic variation and disease, we can plot the relationship between genetic variants and disease in this way. In the y-axis, we have penetrance, which is how severe is the effect of the variant. And then on the x-axis, we have the frequency, how common are variants within our population. So whenever a variant is associated to disease, we can have a pathogenic effect that is mainly populated by rare variation that have a large effect. These are the variants that by themselves can cause disease. These are very rare, they're not established in the general population, but they have a high effect. Then on the other side, we have common variation that contribute a small effect to the disease, any disease, and thus they can be established and be common in the population. These variants are thought to have a collective effect, a cumulative effect. So on this side, on the genetic spectrum, we can start to talk about risk and an ethnic component. Naturally, developmental epileptic encephalopathies and SYNGAP1 variation falls within the realm of Mendelian variation, of rare variants with large effects. SYNGAP1, as you all know, it has a complex presentation, but it has a monogenic mechanism of inheritance. Usually, one de novo variant can cause the disease by itself. It has an incidence of 1 every 16,000 naturally born individuals. And we can expect 400 new cases per year in Latin America, and in the United States, this number, it's a bit lower, is I think 366 individuals, new cases per year. The presentation of the disease is really complex. We can have a lot of comorbidities and symptoms within SYNGAP-related disorders. And these symptoms are complex in a time dimension too. They evolve differently and they present themselves in different ways as long as the time moves. And this is really important for the study of these disorders to manage expectations and see what we can expect from different mutations. It's always really heterogeneous for this type of syndromes. But one important thing to know from the beginning is that not all SYNGAP1 variants will cause the same disorder, but also not all SYNGAP1 variants will cause a disorder by themselves. Most of the genes that have been associated to neurodevelopmental disorders, they have naturally benign variation within them. And it's easy to find variants that do not cause any harm at all. And knowing this, variant interpretation is key. So let's move to the process of variant interpretation. This is a typical genetic test. And from a gene panel, we can see that we have 151 genes that we know that are commonly associated to disease. And then the company tells us a clinical interpretation. In this case, variants, we have a SYNGAP1 variant of uncertain significance. This type of interpretation does not allow a diagnosis. It doesn't mean it's wrong. It's just that it's not useful. And why genetic diagnosis is so important? You know way better than me that knowing the true cause of the disorder ends the diagnostic odyssey, which is a source of stress, psychological and economical stress for patients and their families. Knowing the genetic cause of the disorder allows for better counseling, genetic counseling, patient management, and lately, in some cases, it can lead to definitive solutions and treatments. Knowing what is happening genetically allows you to access support networks such as the Syngap Research Fund, and also to participate in clinical and genetic data aggregations that allows scientific studies to be developed to understand better the disease. The power comes in numbers. Naturally, these benefits from genetic diagnosis have a synergic effect. The access to support networks improves clinical and genetic data aggregation, which in turn allows for more pathogenic variation to be identified, allowing more patients and families accessing a diagnosis and also better counseling and patient management. So it's really important to access genetic diagnosis, to reach a genetic diagnosis. Unfortunately, for most of SYNGAP1 variants, we have 1/3 that it will fall within the variants of uncertain significance realm. And we can see that it's the most populated category between clinical interpretation. Then if we move to the missense variant side, the most common type of variants, the variance of unknown significant rates goes up to more than half of the variants being diagnosed with this state. And personal communication with companies, I have heard that for each pathogenic SYNGAP variant that's diagnosed, 2 SYNGAP variants are identified. But not all is bad news, right? Those can be reinterpreted. And in this matter, what can we do? When a variant is pathogenic or benign, how we can put our effort, our scientific effort to try to improve this procedure of interpretation. What do you think? What is an argument to say that my variant is pathogenic or not? There are several criteria that can emerge. For example, has my variant been tested in a laboratory in a molecular setup? Do I know that the variant caused a deleterious change in the protein function? Is my variant established in the general population with no known effect? Has my variant been observed in other patients? These are all different arguments that arise when you sit on the table and discuss about variant interpretation. So how to interpret a variant and its relationship with disease? Turns out that it's quite similar to how to solve a crime. If we consider the case of Mr. Variant against Mrs. Patient, we have a variant interpretation setup. We have a variant that is a suspect accused of causing the disease of the victim. We have evidence and we have alibis. Of course, the crime is the disease itself, and the judge will be the interpreter, which can be any of us. Of course, within this trial, we have some rules, the penal code. And this was published in 2015 by the American College of Medical Genetics. It was elaborated in agreement by clinicians, scientists, genetic counselors, and laboratories. It consists of 31 criteria for variant interpretation that can be aggregated together to make a genetic diagnosis. These are the criteria. I don't intend to show you each one of them, but it's very important to know that there are some criteria for pathogenicity, that means the evidence, and there are criteria for the neutrality of the variants, which are the alibis in this case. We have strong arguments for alibis, we have supporting arguments for alibis. Not all criteria have the same strength. We have very strong arguments, moderate arguments for pathogenicity, and just supported arguments for pathogenicity. Together, they can help us to make a call and make a judgment about the variant in question. Some examples of strong evidence: a well-established in vitro or in vivo functional study supportive of damaging effect of the gene or the gene product. Moderate evidence, that means in a lower scale of interpretation, is that my variant is located in a mutational hotspot and or a critical and well-established functional domain, an active site of an enzyme, for example, without benign variation. That's also really important. Then on a supporting level evidence, that means even in a lower scale of confidence, we have multiple lines of computational evidence. Here, bioinformatic methods available are plenty. And I will show you a couple of examples later on. Then we have alibis. As I already mentioned, if a variant is observed with a high frequency in the general population, it's unlikely to cause a severe syndrome such as a developmental epileptic encephalopathy. NOMAD is here the name of the database that aggregates variation from the general population. It's not the only one. There are several. Then we have supporting alibis, in-frame deletions or insertions in a region known to be repetitive in different configurations and without any known function. This is also an argument of supporting alibis. So having all these criteria and arguments in place, is my variant guilty or innocent? Naturally, we have two states, a pathogenic state, a guilty state, and then a benign call, when my variant is innocent. But variant interpretation is a bit more complex. It's not binary. We have 5 different levels of clinical significance. From benign, we have likely benign. Then we have uncertain significance in the middle when I cannot make a call for each side. Then likely pathogenic and pathogenic. How is the variant called? Well, the ACMG suggests a method for different criteria combinations that allow us to make a call. One very strong argument alongside a strong argument will allow us to make a pathogenic call. Then, only in the absence of benign arguments, 2 moderate arguments alongside a very strong argument will allow us to make the same call of pathogenicity. On the other side, if I find at least one or two supporting arguments for benign, I can make a likely benign call, also in the absence of pathogenicity arguments. In any case that these configurations are not possible to make, we will be obliged to make a variance of unknown significance result. And that is, all other cases will be called as unknown significance, which explains also why this category is so populated. It's so populated in patient repositories and clinical laboratories. Some considerations about the interpretation. Different interpreters might reach different conclusions or even opposed conclusions. The information required for each criteria is difficult to access. Making a molecular experiment, a functional experiment on one variant, it's usually really expensive, and not many people can do this. Then information available evolves constantly, which makes reinterpretation possible. And it's very important to keep in mind that these are guidelines, not rules. So it's up to the judge at the end to make the call. Coming back to all the 31 criteria, I would like to highlight the computational methods available here, which leads me to the complementary methods. Bioinformatic criteria are considered among the ACMG guidelines. There are some strong computational criteria. Let's say if I find my variant in patient variant repositories, I can match my variant on my hand and see if it has been observed in other patients. Then we have other computational methods related to frequency, to mutational hotspot, and then multiple scoring that I'm going to describe to you now. Basically, bioinformatics methods can be separated into categories. The first one measures biological context. That means measuring the effect on the chemical exchange observed. If an amino acid changes with another that is similar, it's not very damaging if it changes completely in structure. And we have structural damage. Conservation is my variant falling within a site that it's conserved between species. This tells us that the site is important if it is conserved. We also have meta scores that combines different scores and thresholds to generate a single score of pathogenicity, and also machine learning associated methods. Here in each of these tiles, I show you some popular bioinformatic methods to score variant pathogenicity. That's just one category. The other category is frequency evaluation, when I use bioinformatics databases to carry my variant and see how it presents itself in the general population and also in patient variants repository, as I already mentioned. Both of these categories come together in the variant interpretation process, and we can use several multiple sources to annotate variants. In the laboratory, and as part of my research, alongside with Dr. Lau's lab, we have developed some specific methods for variant interpretation. But also we lately realized that it's not only about generating fancy scoring pathogenicity methods. We also need to understand what is happening and make the information available to a broader audience. And for that matter, I wanted to also highlight simple Climber, which is a method to reanalyze and see how patient variants fall within the gene and put the information contained in a large database in a easy to use context. This is how patient variants look on Climber. We usually get access to a big table with a lot of information that is connected to multiple other databases. But if you're not close to the bioinformatic or genetic fields, this table is difficult to digest. I mean, the HGBS format of variant calling, you usually get the result from a clinical genetic test company is difficult to understand. It's not straightforward. So for this, we developed a database called Simple Climber, which I invite you to explore, especially for Syngap. And the tool grabs the entire database of Climber and lets you ask more questions than the ones that are possible to make in the original database. It's the same information, but make it in a more easy to digest way. If you type Climber here, or if you type Syngap, you will see a summary statistic of all the variants that fall within the gene. You can automatically map the variants that are known to be associated with Syngap-related disorders. We can see here there are a lot of them reported. The latest release already contains 680 variants from SYNGAP1. And it's something that you can use to explore, to compare your own variants and see if there are some trends there. Of course, the tools that we have developed are not the only ones. We have a lot of resources available in the internet. I can share with you internally all of these sites. There are a lot of resources lately to analyze variants. So this is a world that it's on development. So we have a lot of resources to be integrated here. Especially just in the last couple of weeks, we already had a report of all the protein structures available, solved by this AlphaFold supercomputer. So it's a lot of studies can be performed right now that may aid in the variant interpretation in the future, in the near future. So now I'm going to show you an example of how to interpret a single variant. Again, this is just an example, idealized case. But coming back to the example genetic test, we have a variant of unknown significance here and a clinical interpretation provided by the company. We have the variant here in the HGBS format that I was telling you about. And then I will suggest to use the judge assistant, which is called the interbar tool. And it's designed to aid in the variant interpretation process. This is an online tool developed by the Wang Lab. It's easy to use and makes all the 31 criteria semi-automatic. So you can fetch all the databases available in the internet in just one place and evaluate how the 31 ACMG criteria behaves on a given variant. We can input our variant from our own genetic test here and see what happens. At the beginning, we get a replication of the clinical interpretation. But then if we go to details and adjust, we can obtain all the criteria and see what we can modify according to our own information. Here, for example, after studying the variant, we realized that first, these variants fall within a gene that has a low frequency of missense variation, as we can see here on the NOMAD database. In the whole database, we can expect nearly 800 missense variants, but we only observe 351. This tells us something, that the gene doesn't tolerate variation in the general population, which means that it's likely to be associated with disease. This is a supporting argument according to ICMG criteria. So we can check that out. Then we know that multiple lines of computational evidence support variant pathogenicity. I have selected here some popular scores that point into the direction of pathogenicity. These are not the best scores. These are the ones that we usually use, and they work really well in neurodevelopmental disorders. But we can also check this out because multiple computational approaches are telling us that the variant might be pathogenic. So with these two arguments, we can reinterpret the variant and we will obtain a likely pathogenic variation, the same variation obviously, but with different criteria included here because I, as a judge, have decided to include these former two arguments. But this is not the only thing that we have in place. We can study the parents, this is available, and we can determine the inheritance mode of the variant. If it is absent from both parents, we can conclude that the variant is de novo. And being de novo is a strong argument for pathogenicity, so we can check that out. If we have the resources and the know-how, we can also make experimental models of my variant in vitro or in vivo to support the deleterious effect of my variant. If I have access to these or to repositories of experimental results, I can check this out too. So we have two strong arguments. If we modify this on the platform, we will get a pathogenic interpretation of the variant, this time with more arguments of pathogenicity. Then, no more question, Your Honor. My variant is pathogenic. As a summary of the variant interpretation process, I would like to highlight that the interpretation is a continuous process. It does not end once I make the call because new information is always appearing. It's bidirectional. If I find a likely pathogenic variant today, it might be a boost in the future because new information arises. So it's highly recommended to not consider the call, the initial call, and especially the genetic test call, a final call, the clinical significance of my variant. Here, keeping up with the literature is crucial. Of course, each case is different, and it's highly recommended to be conservative, innocent until proven guilty, right? And this is really important because taking light in the pathogenicity call has some strong effects. First on the environment, because if I make a call on a variant that is pathogenic, it will stay there and it will be used as a reference for future calls. So it's really important to be conservative, and be entirely sure of the call that we're making. If not, then it's better to make a variance of unknown significance call. Of course, the interpretation is better in a multidisciplinary setup in collaboration. Interpretation requires clinical, molecular, and genetic expertise. It is not a standalone process. And then also, I would like to highlight that the adequate interpretation allows also accurate candidate selection for clinical trials. And we all know that this is gaining importance as new therapies are being developed. So as concluding remarks for this talk, I would like to talk to where this field is going. We all know the complexity of neurodevelopmental disorders, and until now I have only talked to you about two states of a possible variant. A variant can be pathogenic or benign. However, all of these syndromes with different severity and stages and comorbidities are all caused by pathogenic variants. So we need to go beyond the pathogenicity call. SYNGAP1 is one good example of the complexity of neurodevelopmental disorders, and pathogenic variants is not enough. We need to know more. We need to know how these comorbidities are related, how the syndrome evolves within time. And genetics can help in the study of this. First, beyond pathogenic, we can predict functional consequences bioinformatically by training machine learning methods. This is an example of a study that I had the privilege to participate, the story of Henrique Hein, where she developed a prediction model for gain or loss of function for sodium and calcium channels. When this has very strong effects on the therapeutics and treatment that this patient can have. So it's really important to know the functional effect of pathogenic variants. Then phenotype prediction. Some variants may lead to different phenotypes, severe phenotypes, mild phenotypes. This is seen in the SCN1A gene when we can have severe Dravet syndrome or mild plus epilepsy. By studying more than 1,000 patients, we were able to model the pathogenicity of variants and develop a prediction model that was able to tell us the percentage of developing Dravet syndrome or GEF+. And this is achieved just by data aggregation and statistical methods. We created a tool to provide any person with a SCN1A variant to develop a prediction for their own purposes. And the other really important issue here is data aggregation and diversity. I show you here the map of the EPI25 consortia, the biggest genetic collaborative to study epilepsy. And we can see here the source of patients that are being studied, currently more than 25,000. We can see here a clear gap that I'm trying to bridge, but the study of underrepresented populations in genetics may shed some light into the genetic mechanisms of the whole disease and also to uncover new mechanisms. So it's really important, and we're all part of this puzzle. So finally, I would like to end with Mike words that pretty much summary what I would like to leave with you. Don't stop when you see a booth, when you have a booth in your hands. Keep testing and keep asking. With that, I would like to say thank you to all the people that I worked with, especially to Dennis Lal, and also to Lisa Marie, who developed this nice allegory of the judgment, the trial.
Thank you, Eduardo. That was great. I think we have questions. I have a few questions too, but Victor is asking if you can see the Q&A. What does it mean when a variant is determined to be a bus based on a low Probean score?
What does it mean for a variant to be?
Yeah,
sorry, I cannot hear.
Do you see the Q&A questionnaire? Are you able to?
Here.
Yes.
Let me. Oh, I'm sorry, I do not know what it's Probian score. I haven't heard of this. If somebody can help us out in the chat with the Probian score.
I don't know. JR, you know what is Probian score? I don't know either.
Yeah, but none of the scores, especially the bioinformatics ones, are definitive scores. Scores can be integrated to the ACMG guidelines as computational evidence, but they do not have the power to determine pathogenicity by themselves. That will be my comment for any score.
Okay, and I have a question. How often, I know we talked before that initially when the companies start doing the genomic testing, there was a lot of positive results. And I want you to explain all that to people. And when is the cutoff when everybody was like, wait, you are doing too many diagnoses, and why that happened? I want you to give a perspective what happened to people.
Well, if you take a look at the rates of pathogenicity delivered by genetic test companies at the beginning, it was high, and mostly because there was a positive pressure to establish clinical genetic testing as a field, as something that it's useful. I don't think this was intentional. It's just how it happened. Then, as we went into increase the volume of variants and clinical variants analyzed, people started to be more conservative, because initial pathogenic calls were not that pathogenic when you started to analyze more patients. And that is one of the reasons that all the stakeholders gathered together and established these ACMG guidelines to make the process systematic. And most companies today, they try to get close or even beyond the guidelines today to be more precise in this process.
Okay, and then the other question is how often you get second opinions of, I mean, how that works, because we know we have patients that come to our, to Syngap saying, you know, I have a bus for Syngap. The kids look similar to our Syngaps, but still they are in a bus diagnosis. How often you reevaluate or to recommend to reevaluate for both, and how do you ask for a second opinion to a higher institution like yours that have somebody who work in these very hard second opinions?
Well, I think one of the most important things is to have access to genetic counseling after a genetic test, because most of the process done by clinical testing are automatic. I know they provide most of the times a report evaluated by a scientist, but still the genetic test is fixed in time. It cannot update itself because it's a paper, right? And you may stay with that, and I do not recommend that. I would recommend to revisit the interpretation, I don't know, once a year for BUS. Information is growing and growing exponentially. So reports are getting better and better. And also there are the tools to study, and I've seen how experts and how fast parents can become experts in the field. And so you are able to do your own interpretation with all of these tools that are available. And the community is quite active. You can always ask some scientists that it's researching the gene who have the molecular expertise to make some calls, to modify some criteria. And I also recommend that to reach out to scientists who most of the times love to help and love to contribute to things that are quite important, right?
Have you seen diagnosis that you have revert from pathologic to benign or possible benign? Have you done that?
I haven't done it myself, but I've seen it. It's mostly because when these general population databases, they have like a new release and they report like twice the size, you start to see variants that wasn't in previous database, but now you see them. And that's a strong argument for benign variants. So that has happened.
Okay. JR, do you have, oh, I think we have more. Victor and Jazz are asking more on the Q&A. And I think, yeah, JR may have some more important questions.
Yeah, well, I wanted to talk a little bit more about Victor's second question. So he had said that so Provian is one of the, I think it's at the Ventnor Institute. It's just one of the tools you can use to look for how different you think the amino acid is, basically. So it seems like for his questioning, the idea is to go back maybe once a year and continue looking at continue inquiring about the VUS with the geneticist or the genetic counselor. Yeah. So a year sounds like a long ways away, but we can also do some of our own research on it and try to bring this information to them. I also think make sure we're in Citizen, because they have a really top-notch genetic counselor in Ellie Brimble, and she, as far as I know, she personally looks at each variant and tries to figure out what's going on. So I guess that's maybe the opposite, because you're supposed to know that you have a pathogenic to go to Citizen. But I think my question for Eduardo, which might have to do with Victor's question, I'm not sure, is how much weight is given when you have a small number of genes on the test. So say you're doing an epilepsy test, an epilepsy panel with just 151 genes on it. Is there the same weight given to that as when you do an entire exome? Right, because if you're only looking at 151 and you have a VUS, what about the rest of the genome that wasn't looked at? I'm just wondering, how does that have interplay in how these are scored, or no?
Well, gene panels evolve constantly. The literature reports a new epilepsy gene every month, if not. So these panels are constantly changing, and it's a source of delay in the diagnosis process. Because when we have the exome, the complete exome, we can do two things. First, we can interrogate all genes possible, and then we can revisit our exome when time passes and see, hey, we have a couple of genes reported. Has my variant been there? So exomes tend to be of use for a longer time than panels. Of course, the panels, they harbor the relatively high chance in a cost-effective strategy. And what I've seen also is that patients who have a negative panel, they go then to exome sequencing, and this introduces a delay in the diagnosis with all the effects that this has. So it's really important to achieve diagnosis fast. And for that, I will always recommend, if you have the access to do an exome. Exome does not provide all the answers, and there are several ways of identifying pathogenicity that are outside the exomes, and it's part of the strategy. I've
right.
seen.
So you're talking about also copy number variation and karyotyping, other tests that have other, and even, I guess, mitochondrial testing. Yeah.
Yes, poison exons, structural variants, repetitions, short tandem repeats, all of these type of variants are not that easy to see on panels or exons. So if you have a VUS on the exome, does that not mean that you don't have a pathogenic variant. There are other tests that can determine alternative mechanisms that could be happening, but we are just not seeing them.
Yeah. I have other questions, but I think maybe, I saw Victor and Jess are both here. Marta, do you want to have them talk first? I'll ask when they're done.
Yeah, Victor, do you want to ask your question?
Sure. Thank you very much for the talk. It was really, really helpful. And I'm a parent. My son was diagnosed with SYNGAP1 based on a VUS, but with like his medical history and the way he presents, he was diagnosed. So this has been super helpful. I only understood a little bit of your talk, I think, but it was,
I'm sorry.
No, it's okay. I've been trying to learn it on my own, and it's definitely a complicated world. And I actually work in medicine too, so I have some of a head start. But my question was around like where should we focus as like a patient group in terms of helping advance the science of classifying variants? I know we're enrolled in like this All of Us, which is like a US based population cohort where you contribute your blood samples and they do genotyping and I think whole exome sequencing, and then you also contribute all of your electronic health record data and surveys. Do you see those as being helpful for this, or is it really we should be doing things like the Citizen and things that are like really like more focused on like rare diseases, I guess? And what's most helpful?
Well, there is a limit in what you can do on a genome-wide level to determine pathogenicity. Not all genes are the same. Not all DNA segments behave the same. So I think moving into the gene-wise direction it's a good strategy, and for that, data aggregation is key. We need to accumulate data to extract meaningful information. The more data and patients we have, the more follow-up we do, we can understand better how these variants behave in multiple individuals over time. So as a group, I think a good strategy is to accumulate numbers and data. This will help us also to develop more robust statistical models specific to certain phenotypes.
And for us, that's by joining Citizen, right?
Yeah, that's the major work we are trying to do now. Is gonna be phenotyping all the genetic data that we have. That's the short-term goal. The long-term is different, but the short-term goal is to get the phenotyping of all those variants and group them. That you are doing a lot of that. Jar is doing a lot of that. That word grouping and everything. But yeah, we are that's the goal, that early goal.
Are you able to enroll if you have a VUS into Citizen?
If you have a VUS, we have, I think, they are like more than 10 right now, and they
Victor said he had a diagnosis though. He had a diagnosis plus the genetic VUS, so that's yes, you should enroll. If you have a diagnosis of SYNGAP1, you should enroll for 100% sure.
Okay. If I'm able to complement my previous answer, data aggregation is really important, and also molecular studies. The molecular characterization of genetic variants found in patients. I think this is key, and also every functional characterization stays as a valuable information for the future. So it might be expensive to characterize every possible variant, but every little step that we make in that direction will help to get the full picture of variability in SYNGAP1, for example.
So can I ask you to elaborate on the molecular characterization? You're talking about like biochemical assays in vitro. You're talking about mouse models.
Yes, mouse models.
Any kind of characterization, whether it's in vitro, in vivo. Okay. Yes,
and any kind. And for SYNGAP, we were discussing before the talk that it's quite complex because of its function, interactions, location. It's quite complex, but every insight that we can make into that direction is valuable, and it will serve the future.
Yeah. Thank you. So Jess had a question, Marta.
Yeah, go ahead, Jess.
Hi, can you hear me?
Hello,
Yeah.
Jess. Yeah.
Hello. So my question is, with the reinterpretation of the bus after a period of time, from my experience, it really relies on the families pushing for the reinterpretation in many cases. And I'm wondering if are we converging on a generalized approach to automatically reevaluating variants of unknown significance in the light of new evidence?
I know that some companies, they provide reinterpretation among certain conditions. Make it automatic. I'm not sure if that is possible, because the nature of the new evidence is it comes from so many sources that it requires on the table different people rather than an automatic method. And on this table, clinical expertise, molecular expertise, bioinformatic or genetic expertise are required. So I don't think it can become completely automatic in the near future.
Thank you.
So as a follow-up to Jess's question, you showed us a great website for looking at Dravet that I think you guys run, right? Is that from your lab? Did you establish that?
Yeah.
Yeah.
Yes, we developed that with it's on revision currently.
So are you developing the same thing for SYNGAP1, or are you developing the same thing for all genes? Or is that something that would need to be a funded project to do for SYNGAP1?
We're not developing something in those lines for SYNGAP1. We are working on related sodium channels, because it's we have figured out that it's easy to study them together. So we're working in that direction with SCN2A and other sodium channels, but not on SYNGAP currently. It will be possible with data available. That method was developed based on data aggregation from multiple patients, variants, age of onset, clinical features like that.
Mm-hmm. If we wanted to try to set up a project like that, how would we go about that as a patient advocacy group? How would we figure out who should do that same kind of project for SYNGAP that you're doing for Dravet? Right, because we don't want it. We want to find the actual SYNGAP patients. We want to find the people that don't have SYNGAP, if they think they do. We want to know which parts of the gene do what, and we want to know it all. So it seems like this type of it's almost like a hunting, gathering expedition to find all this data from all these disparate sources and interpret it.
Well, and we started the SCN1 project with one question: Can we differentiate mild presentation of the disease with severe? And if we can do that with SYNGAP, which I think we could, based on the data aggregation that you have, might be possible.
Okay, yeah. So maybe you have a postdoc that might want to do this.
Sure, we could.
Okay, yeah. Okay, we can talk about that more. I don't want to put you totally on the spot about that, but this is something we all need. Like our patient community desperately needs this, and we're all kind of feeling around in the dark. And I'm maybe the leader of feeling around in the dark, but I can't, people are like, oh, I can't do this. This isn't something I can do, but it's something that we all think about and that we need right. Concerted, we need a concerted effort toward it. So it's very exciting to think. Okay, so I want to ask you a question in a different realm, if that's okay. When you showed us the different types of mutations, one type of mutation that wasn't on your chart was sort of upstream, variations in the upstream regulatory regions. So something that might change the expression level. And I was wondering if either for Dravet or for Syngap, if you look at upstream variation, and there's two reasons. One is to want to know about how expression levels, because if half is so severe, maybe less is going to be some sort of mild, right? Some sort of mild phenotype. And then the second reason is, as we get toward precise genetic therapies, there are some times when we might want to distinguish between the two alleles. And so knowing if there's general variation in the population already, that is it likely that we'd be able to distinguish between the two alleles just by normal population variation? Do you see what I'm saying? Not that we know a priori which it was, but you might have to figure out which one is which, but is there variation there that would allow us to distinguish between the two alleles in most humans?
Well, the question has several branches, and it's complicated sometimes to differentiate that something is not there because it's not there, or because I'm using the wrong glasses, let's say, or the wrong platform. For UTRs, structural variation, repetitions, sometimes there is a technical difficulty in seeing those variants to increase our own awareness that maybe we have some Syngap variants that are increasing the expression on a non-coding region, and non-coding regions are not tested in most of the clinical genetic testing approaches. So that's a technical difficulty. If we have the resources, we will test the whole genome and we will explore everything. And even that will not be enough to detect all pathogenic events. And that we know that there are at the transcriptional level some errors that may lead to gain or loss of function of certain genes. So it's really important to be aware of the methods that we have. Unfortunately, once the test is done, it's not possible to revise things that are not read. So it might be difficult to evaluate that.
Okay, so in the epilepsy gene panels, they don't look at some of the upstream, because I thought in whole exome they lived at least a little bit outside the exome.
Sure.
Exons.
Sure. In the whole exome, we have some proofs that detect UTR. You can even design your own proofs to scan those regions more in deep. But we're still learning how genes are expressed. So that's also something that we need to consider. The poison exons are a good example. They're difficult to detect in exome sequencing because you don't have the proofs, because you weren't aware that these portion of the genomes were actually exons that appear suddenly. So we also have regulatory regions that are far away from the gene itself. So these regions in trans that regulate expressions and some EQTLs. It's not entirely reduced to the segment of the gene.
Okay, well, I of course asked something that can't be answered, so sorry about that.
No, not at all. It's an important discussion to discuss different mechanisms.
Mm-hmm. Yeah, I was part of a discussion with some people from some ring chromosome diseases, and they were saying that their diagnosis rate is going down because people don't do karyotype analysis as much anymore. So as things being diagnosed with panels and whole exome are going up because of the increased testing, their diseases are being less diagnosed. So I found that interesting that the things that we've known for a long time are starting to not be diagnosed as much, just because the tests are falling out of favor. Okay, so it looks like Mike would like to answer some questions live, so I'm going to stop talking now.
I think we answered all the ones that were in the Q&A, but yeah, I think all that you guys were talking about, it goes to the relevance that for probably people that has bosses, going to the genetic counselor once a year probably is very valuable, because that's the problem. This is a science that is just beginning. It's a lot to know, and a lot of things that probably will come up in 1 year, 2 years, 3 years, 5 years. And for the ones we have the pathogenic diagnosis, we probably will be smart at least every 3 years, I think. What do you think, Eduardo? I think revisit the diagnosis is probably important.
Yes, for sure, for sure. UES are must be so frustrating for people having that test in their hands, and it's a responsible claim to say that reinterpretation might give you hope. If not now, maybe in a year or so, we might, as the science community, accumulate enough information to make the call.
Yeah,
So,
another thing is, for example, I have Sophia, my daughter, has the pathogenic for SYNGAP, but also has 2 bosses in her genetic report. And there is something now that is coming to evidence in the genetic that everybody has a different background too. Then how they will show the symptoms also depend on the genetic background as a whole.
right. Yeah, that's also really true, because you have causing factors, but you also have modifier factors that are maybe not entirely related to SYNGAP1, but just the background, as you say. For that matter, I'm also really interested in gathering information from underrepresented population, or also people with mixed genetics, like admixture, that it's so common in Latin American populations. That background might explain why some patients react differently to drugs, or manifest different disease progression, or other rare variants within them. So that's really, really true.
Okay, I guess any other questions? I think we are at the end of the hour. Any questions? Or it was great. Thank you so much, Eduardo. And this is, I think, very clear and very helpful for a lot of families. Thank you, and I hope Santiago gets a little bit warmer to
Thank you.
weather anymore, and we truly appreciate very much you are doing this for us.
Thank you. It will
Hope to stay connected, and like JR said, we were talking before about working together, and we are still thinking about it. Yeah, we are trying to do all these projects that are very important, and of course your project is very important to us.
Thank you so much.
Thank you so much. It's truly wonderful. Thank you.